Hi everyone. So I hope you can see me, quite short, but I'm Bina. I am currently a research associate and a neurology doctor working in the Caroline Williams Gray Lab at the University of Cambridge. And I'm a sub-investigator on the DAPA-PD clinical trial, which has been funded by the Van Andel Institute and Cure Parkinson's. Over the next 20 minutes, I hope to tell you a bit about the background and rationale for the trial, but also tell you about the trial design and setup. As I'm sure many of you are aware, the core pathology in Parkinson's disease involves the loss of dopaminergic neurons, so these specialized nerve cells in the part of the brain called the substantia nigra. And with the loss of these nerve cells, you get a loss of a chemical called dopamine, which is what they produce. And as a result of that, you develop the key symptoms of stiffness, slowing, and tremor. With progression of the disease, people can develop a whole host of other symptoms, which are not necessarily attributed to the loss of the nerve cells in this particular brain region. And we have a lot of treatments that can help with the symptoms of disease, but we don't have any treatments at the moment that can slow or stop the progression. A lot of research is now, as highlighted, trying to find drugs or medications that can help slow the progression of the disease. But the main question is, why are these nerve cells being lost in the first place? And if we know why they're being lost, then we can target those mechanisms. And as you might be aware, there's a lot of research out there with lots of different proposed mechanisms of action. But the one that I am particularly interested in and working on is the thought that inflammation and immune system activation contributes to the progression of Parkinson's disease. So our immune system is our body's defense against infection or injury. And there are two main components of our immune system. We have the innate immune response and the adaptive immune response. Our innate immune response are the first responders, so they get there as quickly as possible to any injury or infection. And it's made up of a variety of cells, natural killer cells, macrophages or neutrophils that can release a variety of proteins that can help defend our body against infection or injury. They can also help recruit and change the adaptive immune response, which is a response that comes later on down the line and is more specialized to any injury that's occurred. This consists of B cells and T cells for short, and they release things like antibodies, which can specifically target any infection. In health, we need a good balance of immune activation against immune suppression. And in many conditions, but also in Parkinson's disease, it is thought that our immune system is slightly overactive and that we have a dysregulation of this immune system. And this contributes to the progression of Parkinson's disease and that nerve cell loss. So there's a lot of evidence that supports this theory. So this is just a subsection of the evidence from predecessors in the lab that I'm working in. And the image that you see there are specialized brain scans of people with Parkinson's disease. And the scan itself uses a radioactive tracer, which binds to areas of inflammation or immune cell activation in the brain, called microglial activation. The hotter colors, the reds, the yellows, are where there is evidence of immune cell activation. And the top row are healthy controls, and the bottom two rows are people with Parkinson's disease. And as you can see in the bottom two rows, there are greater levels of this inflammation in participants with Parkinson's disease compared to controls. And this inflammation or immune activation isn't just localized to the brain. You can also see it in the blood. And looking at this kind of bar chart here, all it does is it compares people with Parkinson's disease to controls. And there are certain levels of those cytokines, which are part of the immune response, that are higher in people with Parkinson's disease in comparison to controls. Thanks also to all the kind people that donate their brains with Parkinson's or without Parkinson's disease. We've been able to look at the brain directly. And what we can see is, similar to the imaging studies, there's evidence of immune cell activation within the native cells of the brain, so the microglia, but there's also evidence of cells that are normally found in the periphery, so in our blood, those B cells and those T cells that have then crossed the barrier into the brain. This is then showing that this inflammation is in the brain, in the body, and there is a mix between the two as well. So this diagram essentially a simplified version of what we think about inflammation and immune activation in Parkinson's disease. What we think is happening is that our nerve cells are degenerating. They can release these alpha-synuclein, misfolded alpha-synuclein, which can then trigger the activation of immune cells in the brain, so these microglia. This can then release cytokines, which can cause kind of a vicious cycle of more nerve cell loss. But also these kind of misfolded alpha-synuclein can talk to the periphery, so our blood and our gut. And all of these areas just represent that everything is essentially talking to each other, and inflammation is present in Parkinson's disease in our brain, gut, and blood. What I'm going to focus the rest of this talk on is a particular part of the inflammatory response called the NLRP3 inflammasome. So this is part of the innate immune response, and the NLRP3 inflammasome is a group of proteins that kind of come together and can kickstart inflammation. And that can happen both in the brain, in the microglia of the brain, but also in the cells in the blood, in the periphery, and that happens in everyone. But in Parkinson's disease, there is some evidence to suggest that there is an increase in the activation of this NLRP3 inflammasome in both the brain and the blood. So as I mentioned, this is kind of a diagrammatic representation of the inflammasome. And the inflammasome consists of a group of proteins that oligomerize or join together essentially when faced with infection or injury to create what we call the NLRP3 inflammasome. And this creates a platform for the activation of certain inflammatory molecules, so some of those cytokines that I was talking about earlier. And in Parkinson's disease, this is again not research that's been carried out by our lab, but research that's been carried out by a group that has shown that in post-mortem tissue, so again, brains from people with Parkinson's disease compared to controls, there is evidence of these markers of inflammasome activation, so these same proteins that are here, that are higher in the brain of people with Parkinson's disease compared to controls. And that's in regions of the brain that are particularly important, so the substantia nigra in Parkinson's disease. And these kind of colourful pictures essentially show that the areas where this activation occurs is in the immune cells of the brain, so the microglia. Not only have we found that there's an increase in activation of this specific NLRP3 inflammasome in the brain, there is also an increase in activation in the blood of people with Parkinson's disease. Oops. And again, this data just shows that people with Parkinson's disease had higher levels of proteins associated with this specific inflammasome. And the higher the level of and higher levels of cytokines that this inflammasome activates, and the higher the level of these cytokines was correlated to more severe disease in people with Parkinson's disease, suggesting it might play a role in the progression of Parkinson's disease. In animal models of disease, as you know, animals can't get Parkinson's, but we can inflict Parkinson's disease in animals using numerous techniques. But in animal models of disease, these are just the brains of mice or rodents that have had Parkinson's disease induced on them with alpha-synuclein. And as you can see, if you give them Parkinson's disease using toxins or other methods, they get a loss of nerve cells in the brain, similar to what humans do. However, if you give them a molecule that inhibits the NLRP3 inflammasome, that loss of nerve cells doesn't occur to quite the same degree. So that level of brown, which is the nerve cells, is nearly the same as mice who have not been treated or not been given Parkinson's disease, suggesting that if you stop the NLRP3 inflammasome, that could be a potential target for Parkinson's disease. Obviously, we can't extrapolate a lot from animals because they're not as complex as humans, but this is the preclinical evidence. So there's a lot of, Dapansutrile is a novel drug that's been created by a company called Olatec in the United States. And what it does is it stops the joining together of these proteins to make the NLRP3 inflammasome. So it stops the activation of these downstream cytokines and hence should reduce inflammation. It is an orally active drug, so it means that we can take it by mouth. And it is thought in preclinical work to cross the blood-brain barrier. So it's thought to get straight to the brain and try and help with inflammation there. It's a completely novel drug, so it's not yet licensed in any medical condition, either in the UK or worldwide, and no NLRP3 inflammasome inhibitors are. However, it has been studied extensively preclinically, and again, using mice models of disease. This first line is healthy mice. This second line is mice in whom they've given a toxin to induce a Parkinson's type model. And the third line is these mice who have been given Dapansutrile, the drug. You can again see that the levels of brown neurons, there are more here when the drug is given compared to when there are not. So showing that dapansutrile might reduce the nerve cell loss in the progression of Parkinson's disease. In these mice, they also had better motor outcomes. But again, these are mice, they're not humans, but this is the best we have. In humans, dapansutrile has been studied in healthy volunteers, in people with gout and people with heart failure for short periods of time. So it has been shown to be safe for those couple of weeks with no severe toxicities or no severe side effects noted. But again, these are still early phase trials which have only been for very short treatment durations. So what are we aiming to do? So as I've briefly shown you, the NLRP3 inflammasome is activated in Parkinson's disease, and dapansutrile, this novel agent, can block the NLRP3 inflammasome and is potentially a novel therapeutic agent for Parkinson's disease. So we're in the process of setting up a trial called the DAPA-PD trial, which is a trial to test the use of dapansutrile, which inhibits the NLRP3 inflammasome in people with Parkinson's disease. It's going to be run at a single site at the University of Cambridge, and Dr. Caroline Williams-Gray is the principal investigator, and I'll be working as a sub-investigator. And it's been funded by Cure Parkinson's and the Van Andel Institute. The trial is a small trial because it's an early phase trial using a novel drug that's not yet licensed for any condition. And we aim to recruit 36 participants. It will be a randomized, double-blind, placebo-controlled trial, which essentially means that 24 of these participants will receive the active drug and 12 will receive placebo to try and nullify the placebo effect or the known placebo effect of medications. And that will last a total of 12 months. Neither the treatment team or the participants involved will know which they're taking, whether it be dapansutrile or placebo. After that 6-month trial period, there is going to be an optional open label phase where all 36 participants have the option of continuing and will then definitely receive dapansutrile. And after the 1-year period of the trial, there will be a 4-week follow-up check just to check that people are okay and haven't had any significant side effects. I'll go into kind of more of the study schedule later on. So it's a very small trial, and like I said, it's a new drug. So the main outcome measure is going to be the safety and tolerability of the drug. So the way that we measure that is by looking at the number of side effects or the number of adverse events that we see in the first 6-month period of that study that I've shown you, because that first 6-month period is mandatory. As it's a short trial and it's in a small group of people, we won't necessarily notice any clinical changes, but we can maybe see some changes in inflammation in Parkinson's disease. So we are going to try and measure different types of inflammatory biomarkers. So the first will be through those specialized brain scans, which I showed you briefly on the first slide. And people will undergo a specialized brain scan at the beginning and at the end of the trial to see if this drug has reduced inflammation in the brain. And the second will be looking at inflammatory biomarkers in the blood and the cerebral spinal fluid through lumbar punctures. This part of the trial is optional, but the blood tests won't be. As it's a novel drug, and I said that it does get into the brain, but that's only from my studies, we will look at whether dapansutrile enters the brain through looking at CSF levels of that drug as well. But that is, again, optional. As we do with every clinical trial, we'll have some exploratory outcome measures, which will be the clinical outcomes. So we'll be looking at changes in motor severity or changes in people's movement, changes in cognition or memory and thinking, and changes in mood, gut function, and quality of life, and other non-motor symptoms. But this is all very exploratory, as the trial is not necessarily powered to see any effects in these. However, if the trial is successful and positive, that could, lead us to further phase 3 or bigger trials. So the inclusion criteria are hopefully quite loose. So we'll be enrolling anyone between the ages of 50 and 80, inclusive. And they have to have a diagnosis of clinically established early Parkinson's disease, which is just using set research guidelines on a diagnosis of Parkinson's disease, but that's fairly inclusive, and anyone with a diagnosis of Parkinson's disease, most of them fulfill those criteria. We're aiming to get people who are either drug naive, so not on dopaminergic replacement therapies, or on a stable dose of dopaminergic replacement therapies for at least 3 months. Because it's a 6-month trial, if people have differing levels of dopaminergic replacement therapies, it could then alter our results. So it helps to keep that bias levels low. And we're hoping to recruit people with an early disease, so within 5 years of diagnosis. Again, this is because research suggests that inflammation and immune activation is an early process in Parkinson's disease. So if we try and get those with earlier Parkinson's disease, we're more hopeful that we'll have a better outcome. And as you all know, Parkinson's disease is very heterogeneous, so it's different in different people. And the pathology or the cause of that nerve cell loss is again different in different people. Because it's again a small trial and we're trying to get the maximum outcome we can, we're going to try and recruit people who have an inflammatory phenotype. So that means people in whom we think inflammation might be playing a role in their Parkinson's. There's no standardized way of doing that, and there's no clear biomarker at present to look for inflammation in Parkinson's disease. But we've used pragmatically CRP, which is a common blood test that's used in lots of medical conditions, and using a cutoff of more than 1 to show people that might have a slightly more inflammatory subtype of Parkinson's disease. And looking at our current cohorts, that's about half of the cohort. This is just some of the exclusion criteria, but essentially we're trying to rule out confounding factors. So anyone that's on any other drugs that suppress your immune system, even over the counter, or if anyone has any infections or autoimmune or inflammatory diseases, or even recent cancers, because that can disrupt your immune system, and inflammation won't be eligible for the trial. Because we're really concentrating on these inflammatory biomarkers, anyone with any contraindications to having those specialized scans will also be excluded from the trial. And also anyone who can't take the medication because it's only available in an oral form. So this diagram is just going through the study schedule. This is the first part of the trial, so the mandatory part of the trial, the 6 months of the double-blind, randomized, placebo-controlled phase. And as you can see, there are a lot of appointments or a lot of visits. At each visit, all participants will see a trial doctor, and they might also see another researcher if there are other tests that can be carried out by someone else. The trial, before you start the treatment medication, you'll come for an imaging visit, and that will be with that specialized scan to look for inflammation in the brain. And then at your first appointment, we'll take blood tests to look at those inflammatory biomarkers before people have been started on the medication, and an optional lumbar puncture, again, to look for inflammatory biomarkers. Then people will have a multitude of safety visits and a multitude of inflammatory biomarkers in the blood. And they will also, sorry, there is an error there. They're not going to have 2 CSFs, that shouldn't be there, it's just here. And they will also have multiple safety checks. So at all of these appointments, people will have an ECG, vital signs and blood tests, just to check on the safety of the drug. At week 4, we'll be doubling the dose of the drug. So between week 0 and 4, people will be on, I guess, half a tablet or 1 tablet twice a day. And by week 4, if they are tolerating it well, they'll be on 2 tablets twice a day. The second part of the trial looks like it has less appointments, but that's only because we don't have as much imaging. We don't have the imaging appointments. We do the same amount of safety checks because some of the participants will not have received Dapansutrile in the first part of the trial, so it's completely new to them. We'll do the same dose escalation at 4 weeks into the trial, and we'll do lots of inflammatory biomarkers in the blood, and we'll take an optional lumbar puncture at the end for those who have consented to this optional part of the study. And again, a multitude of safety checks throughout. So where are we now? We're currently in the study setup phase, and we're hoping to start recruitment early next year. With this timeline, we would hopefully be finished in the next 2 to 3 years. And I just finally want to end with a thank you, first of all, to all the participants who come along to our research clinic. That's helped with a lot of the preclinical work, and also helped with all the clinical trials, but also to the Van Andel Institute and Cure Parkinson's for funding, and Olatec for providing the drug. Thank you.
Thank you very much, Bina. That was fantastic. We do have time for questions. So if anyone has any questions from the audience or maybe online. Oh. Maybe a question from the side. Oh, here we go.
Okay, that sounds better. So I didn't quite follow. What's the fundamental objective of this study? Is it to look at efficacy, or is it to look at safety and tolerability, or is it a mixture of both?
So the primary outcome measure is safety and tolerability of the drug, because it's a very small trial involving only 36 participants. To look at efficacy, you'd need something much bigger.
So I guess my question then is why not do a shorter trial where you just look at open label for everyone if you want to establish the safety first, rather than this sort of hybrid where it looks like you spend 2 years doing it, but you kind of don't end up with full information on either subject, if you see what I mean?
So some of the safety and efficacy, safety and tolerability has already been done in humans. So it's not that it's the first trial in humans. It has been done in healthy volunteers, gout, and people with gout and people with heart failure for 2 to 3 weeks, which is a very short treatment duration, and it's currently in longer duration trials in other conditions. So it's not going to be the first trial of this drug for longer durations. The reason this is safety and tolerability is it's the first one in people with Parkinson's disease, which is a slightly different population. And I guess the rationale for it being a placebo-controlled kind of double-blind trial is that if we see changes in those inflammatory biomarkers, which we might do at these small numbers, it supports the use of supports us trying to use this in bigger phase studies. We might not necessarily see the clinical outcomes, but we might see the biomarker outcomes.
Okay, thank you.
The neurons was also preserved in lighter.
So with this particular drug, so I haven't done any of the rodent studies, so this is all research done by the group in America that have developed the drug. With this particular drug in Parkinson's disease, so far it's only been published in that mouse model. However, they are undergoing, I think, further testing in different rodent models. Other inflammasome inhibitors or NLRP3 inflammasome inhibitors, I think, have been used in bigger animals, but not Dapansutrile in Parkinson's disease.
Neurons was preserved quite well as was in. Do you know if the neurons was preserved as quite well as was in rodents?
I'm not too sure on the other inflammasome inhibitors in bigger mammals.
Question in the front row.
Hi, I would be interested in potentially volunteering to take part in the trial, but I'm too young. What's the justification of having to be age 50 plus?
I guess I don't fully know that answer because I'm just kind of a sub-investigator, but I think the rationale was try to reduce the chances of getting genetic forms of Parkinson's disease, which is why we go for 50 plus.
Tom.
Thanks, Bina. I'm going to ask you a question that I get asked a lot to see how good you are at answering it. So are you just testing one dose, and which dose and why?
So we are testing just one dose. There have been earlier phase studies that have tested a range of doses, and they've found that the maximum tolerated dose is 2,000 milligrams a day, or the maximum dose that they've used in the past that's been tolerated well is 2,000 milligrams total a day. We'll start at 500 milligrams twice a day, and then if that is tolerated well in our participants at 4 weeks on a case-by-case basis, increase to 1,000 milligrams twice a day. And why is because that's the best dose that's been trialled in the past in the earlier phase studies.
Very good.
So I should just add that Dapansutrile is how I pronounce it.
I thought I was pronouncing it wrong when you said it, so I'm not
No,
sure.
I think I'm pronouncing it
I
wrong.
don't know how to
But
say it.
this is a molecule that was put in front of the ILCT committee a couple of years ago and prioritised, and as a result, we approached Caroline Williams-Gray and colleagues to have a chat with the company and explore the idea of doing this trial. So that's kind of the background from our standpoint. On the back row.
Thanks. Firstly, I'd like to congratulate you on a fantastic presentation. It was so clear, and even I could follow it, and it all made sense. So thanks very much for communicating that. Just a couple of simple questions. Firstly, in the tests on the animals, was there any element of restoration of the brain, or was it stopping the development of Parkinson's?
So I guess I never did those tests on the animals, but in the study that I looked at, they looked at animals that hadn't been treated or hadn't been given Parkinson's, animals that had been given Parkinson's, and animals that had been given Parkinson's and then the drug. So there wasn't a restoration effect as such. It was just that those animals that had been given Parkinson's and the drug lost less of their neurons than the animals that had been given just Parkinson's.
Thanks very much. The second question briefly was interested in the blood test element of it, whether there's room for developing a blood test to test for Parkinson's based on your discoveries.
So I am very early on in my research career, so I don't know much about blood-based biomarkers for Parkinson's, and there is obviously a lot of research out there. At the moment, there isn't a single blood test we can do.
And the third quick question was, those on the clinical trial, do they have to drop any other medication they're on, or can they carry on that and then take the additional medication?
So they don't have to drop any other medication that they're on, no.
Thanks very much. Thank you.
In the middle, in the back row there.
I think we might have an online question after that.
Yes, yes.
Hi, I was looking at the cytokine list from the research, and I noticed IL-17A wasn't up there in the literature. Is this one you'll be tracking? Because it does have an association with the NLRP3, and they're related.
So actually, at the moment, it is not in our primary, as you saw the list, in our primary kind of in our secondary outcome measures. Yeah. It is something we can consider. So you said IL-17A, would you recommend?
I would really recommend it. And also, the drug is highly selective, but also not. It bumps into several kinases in T cells, which would affect cytokine levels from Th2, Th17 helper cells. So I think it would be something well worth looking into. There's literature on it. I'm sure you can find it.
We are freezing down our serum and plasma and PBMC samples, so peripheral blood cell samples. So all of the analysis will be done at the end of the trial. So there is still room for movement as developments and technology changes to adapt what we test for.
Okay. Yeah, because you'll have the blood. You should just be able to check for it. But all right, thank you.
Thank you.
And now we have a question from online.
Hi, I'm just going to kind of combine these because they're about relatively the same thing. So firstly, are you guys only recruiting people that are based in Cambridge? So would you have to travel to Cambridge to participate? And then there were quite a few exclusion criteria. So do you anticipate any sort of difficulties with recruitment? How might you are aiming to overcome that?
So in terms of recruitment, this is a single site study. So we are recruiting only from Cambridge. We can recruit from kind of the wider area, so long as people are willing to travel to Cambridge. There is some travel reimbursement for that, but it is only at Cambridge. In terms of, do we think we'll get any problems with those exclusion criteria? I think they're quite valid and important to keep. And we are only aiming to recruit 36 participants, so I would hope not. We have quite a vast cohort of people at the Cambridge Parkinson's Clinic, and we can expand further if need be.
Any more questions from the audience? Oh, in the second row there.
As far as I know, we don't currently have an accepted measure of progression of PD. So when it comes to doing efficacy studies, how are you going to know whether changes in your immune markers do correlate with progression or not?
That's a hard question. So in terms of whether this treatment is successful or not. I guess the main outcome for our trial is whether it's safe, tolerated, and then does what it says on the tin. So does suppress inflammation as it should. I don't think I know of a way of correlating inflammatory biomarkers with progression unless we see a change in clinical progression. Might have to ask for help from others with an answer to that one. But
I was going to say, given that we've got the expertise in the room here, I'm going to throw this question at Tom, and we'll see how he handles it.
Thank you. So, I mean, there's a lot of work going on trying to get better at measuring progression in Parkinson's disease and thus the response to potential disease-modifying treatments. And we've done a lot of work over the last 3 years, getting the views of patients about how best to do this, and the views of people who are expert at imaging, the views of people who are expert at all the different wet biomarkers in blood and spinal fluid, and also the digital wearables, using sensors or that you can walk around with to measure how your walking is, or to use tapping on a phone, measuring tremor on a phone, these types of things that can be more sensitive at looking at change over time. So we have to get better at getting the data to support what patients say is important to them, so the patient-reported outcome measures, and how those important patient-reported outcome measures can be predicted earlier by an imaging, a wet biomarker, or a digital wearable. So we don't need to follow up so many people for so long before we make a decision about whether an intervention is good or not. So you're right, we haven't got the data. We know, we could recruit hundreds of people and follow them up for years, and then we'll have a patient-reported outcome measure that's absolutely robust, but we want to be more efficient, so get something that's more useful earlier.
One last question from the audience. Oh, right behind you.
This is very naive, but how do you give an animal Parkinson's if you don't know how they get it?
so there are various different models that we can use. So there are toxins that we know that humans who had particular drugs in the past develop Parkinsonism or symptoms of Parkinson's, and extrapolating from that you can give toxins. That's not the best model, but that is a model. And then other models of Parkinson's in animals is where we introduce the misfolded alpha-synuclein, so the misfolded protein that we see. I don't do animal work, I don't know exactly how that's done, but those are models. There are some of the models of Parkinson's.
Very good. Thank you, Bina. Very much appreciated, and a fantastic talk.